Dear students, welcome to Genetic Engineering Lecture Part 2. This is part of the Agricultural Technologies ALM207 subject, which is offered at NMIT as part of the agricultural degree. My name is Dr. Nikki Cooley. This lecture will come to you in a slightly different format to the more traditional lectures. In this lecture we will look at aspects of the genetic engineering technology. This will be presented in either a written or a video context. Please complete the associated reading or watch the video and then complete the exercises. Only upon completion of these tasks will you gain the knowledge required for the learning outcomes, so please note that any of this material may be assessed. Please watch the following YouTube, the link can be found on the screen, of the summary of genetically modified organisms. Please complete the following questions as you are watching the GMO video. This is published by Cardiff University. The first question, what is the most common form of genetic engineering technology? Number 2, in 2009 the USA was the largest producer of GMO crops. What were the two countries that ranked after the USA? Task 3. What was the area of planted GMOs in the USA in 2009? Question 4. What proportion of the world's soybeans are now GMOs? Question 5. List the 6 examples of GMO crops and their associated countries that in 2009 were grown in the world. And finally, Question 6. What are the realistic achievements that the technique of genetic engineering can currently achieve according to Cardiff University? You can please refer to the attachment on Moodle for an electronic version of these questions where you are able to type in your answers. For task number 2 on genetic engineering, you may have to refer to some previous lectures and/or YouTube videos. Please look at the figure on the slide. What is the technology illustrated in this figure? You will see from the slide that there are 6 stages. Please describe each stage. You can obtain an electronic version of this on Moodle and you are able to enter your answers electronically here. The following video is a video about the role of the USDA government-run agency and its role in genetic engineering. Please answer the following questions. Again, an electronic version of this can be obtained via Moodle. Please watch the first video, the link shown on the top of the screen, on how they cloned Dolly the sheep. Then prepare a no more than 100-word description of how they cloned Dolly the sheep. Follow this by watching the second video on the summary of GMOs. This was shown earlier on in the subject. Please list the examples of the genetically modified crops that they describe. Task 5 is a case study based on the biotechnology in China. Please refer to the HTTP web-based link for the article. Then please ensure that you go to the section on agricultural biotechnology. There is a copy of the first few sentences of this paragraph on your screen. Ensure that you only answer the questions on the topic of agricultural biotechnology and do not refer to the other sections on China's biotechnology. These sections will not be covered in this subject. Please answer the following questions. What are the forces driving China's requirement for biotechnology? Please list the crops that China prioritizes for biotechnology funding. Other than insect resistance, what other issues are you able to list that the Chinese are hoping to address with their GM program? The Chinese developed the Bacillus thalassorium resistance in cotton. What method did China's scientists use to transfer the Bt gene into the major cotton varieties? Genetic engineering as a technology is coupled with politics, ethics and morals. Many groups have expressed interest in this technology and its applications. They have put together educational and informational videos. Please refer to a counter-argument on genetic engineering. The link is found at the top of the screen. I wish you to view this video and then have a think about the discussion the video claims to make. For example, one of the claims made is that the insertions of genes is random. Is this correct or not? Ultimately, it depends on the technique used to analyse the genome. Some techniques certainly are random. Techniques now exist to analyse the genome rapidly and cheaply. Therefore, a counter-argument that screens technologies could overcome this issue. Please examine the various statements made in this video. Are some misleading or are they all accurate? Please ensure you identify where scientific references of these claims are made. And this brings us to our final task, Task 7. Please write a summary of the advantages, disadvantages and ethics as you see them on genetically modified crops. You will note there is no direct reading or video link for this task. However, all the information that has been presented to you in this series of lectures can be used to draw on your answers and integrate your own thoughts and discussions.